So good morning friends. So today I'll be just covering a small snippet on this vaccine trial that came on 20th July 2020. So this is an important paper because this really looks into definitive therapeutic option for mitigating this whole frenzy about this COVID-19 virus. So this is a paper which came in Lancet on 20th July. So this looked at so this particular trial was done in 5 trial sites in UK. So the title of this article was Safety and Immunogenicity of Chad OXN COVID-19 Vaccine against SARS-CoV-2. So this trial was done in 5 sites in UK. So what they did was, so they took this chimpanzee adeno-vectored virus, which had these spike proteins incorporated into it from the SARS-CoV-2 virus, and this was compared with a placebo group, which was meningococcal conjugated virus. So these two were compared, and the vaccine consisted of 5 into 10 to the power of 10 viral particles, and this was given as intramuscular injection. So the age of the patients that were recruited into the study was 18 to 55 years, and 10 of the patients who were recruited got first dose, after which 28 days later they got the second dose. So they got a booster dose was given in 10 patients that were part of this study. So they looked at both humoral response and the cellular response. The humoral response, they looked at 5 components. So one is the IgG ELISA. IgG levels were checked by ELISA. They looked at neutralizing assay by micro neutralizing assay, and then they looked at neutralizing assay by plate reduction neutralizing assay, and they looked at 3 virus neutralizing assay and the pseudo neutralizing assay, and they also looked at multiplexed immunoassays. So these were all some of the viral methodologies which I may be agnostic to. So these are the different methods they looked at for the humoral response. So the cellular response was looked at by measuring the interferon gamma with ELISA, and the safety of this vaccine was checked up to 28 days, and they looked at any sort of a serious adverse events that developed. So what were the results? So they recruited 1077 patients in the 5 UK sites, and the comparison was made between this chimpanzee adenovectored uh, vaccine, where the spike protein was incorporated, compared with the meningococcal conjugated virus. So they had 543 in the COVID sort of a vaccine and 534 in the control group. So they looked at the side effects. So when they looked at the temperature, chills, malaise, and headache, they tend to occur more commonly in the COVID vaccine group as compared to the control group, and that was statistically significant. And the authors do mention that a prophylaxis of paracetamol could effectively mitigate these sort of symptoms that manifested in the vaccine group. But when they looked at serious adverse events, there was no major serious adverse events in the COVID vaccine group. So they looked at the T cell response. The T cell response peaked at day 14 in 43 patients, and they had a median of 856 spot forming cells in the mononuclear cells on the peripheral smear. So the peak response happened with regards to T cell response at day 14 in 43 patients. And then they looked at antibodies against the spike protein. I think, friends, we understand that the spike protein is the most harmful component of the SARS virus, and most of the vaccines are looked at in targeting this spike protein. So they looked at the IgG antibodies against the spike proteins, and the peak of these antibodies was formed at day 28 in 127 patients, and the measurement, when they measured it, was median of 157 ELISA units, which was found in the COVID vaccine group. Then they looked at neutralizing antibodies against the SARS-CoV-2 virus, and they found that the neutralizing antibodies against the SARS-CoV-2 virus was present in 91%. So 32 out of 35 patients had good neutralizing antibodies when they measured it by microneutralizing assay. But when they measured it by plate reduction neutralizing assay, the neutralizing antibodies was present in 100% of the patients. And what they also looked at is the 10 patients who got the booster dose. All these 10 patients showed good amount of neutralizing antibodies against the virus by day 42. So in summary, so I'm just covering the gist of this paper because I'm not a virologist to dwell into the whole detailing of all the types of responses they saw, but pretty much the essence of the paper mentions that there was a good amount of neutralizing antibodies, and there was a good amount of anti-spike antibodies, and there was a good amount of cellular response that they witnessed by looking at the spot forming cells in the peripheral smear. I think this is what is the essence of this whole paper. So the conclusions the authors make was this vaccine was found to be safe. So it had a good acceptable safety. So any vaccine trials, I think the key component would be to look at the safety profile of the vaccine. And the paper also reiterates that the vaccine did manifest with good humoral and good cellular response. And most importantly, what they found is when the second booster dose was given after day 28, there was a good antibody response that was found at day 46, day 42, sorry. So that's pretty much is the summary of this trial. So thank you very much, friends, for patient listening.